Hi everyone. We're going to continue our study on more in depth on transcription translation. Okay, so just a broad overview. Transcription is when the DNA becomes mRNA, and then translation is when that mRNA is read into proteins. So we're going to discuss how that actually happens. So remember that we have codons on the mRNAs, which are just 3 letters, 3 nucleotide sequences that correspond to a very specific amino acid. And so this chart is just details all the different sequences you can have and the amino acids that they correspond to. This right here is the first letter, this indicates the second letter, and this would indicate the third letter. And so when you look at this chart, you can notice that almost all of these sequences of nucleotides correspond to an amino acid except for 3 of them. So there's 3 very specific codons that are actually stop sequences, which is what tells the ribosome to stop processing, and it's the end of the protein chain. AUG is also very special. It corresponds to MET or methionine, I think, which is a specific amino acid, but it's also the start codon. So almost all proteins have this as the very beginning amino acid. It's like the beginning of the protein chain. So in terms of cracking this genetic code, how they were able to discover which codons corresponded to which amino acids, there was someone named Nuremberg, and he very cleverly created an artificial RNA that was only made of urocells. And so the only codons possible were U, U, U. And he found that when he put these into cells, that the only protein that was formed was a phenylalanine amino, a protein made of only phenylalanine amino acids. And so thus he concluded that UUU must correspond to phenylalanine. And so this was the first discovered linkage between the codon and the amino acid. And they did a lot of variations of this kind of experiment to figure out the other codons. So something to note, notice that there are multiple codes that correspond to one amino acid. So this is considered redundant. You can have multiple different codes and they all lead to the same amino acid. However, one specific code will always be the same amino acid. So what I mean is you can have UUU or UUC lead to phenylalanine. So that's redundant. You have 2 different ways to get to this amino acid. But if you have the code UUU, it will always lead to only the phenylalanine amino acid. So the code is very specific. So how exactly is RNA transcribed from DNA? So we're going to take a closer look. So this picture kind of diagrams the entire process, and we'll go through step by step. In the beginning, it's called the initiation phase. And you have something called the RNA polymerase. It should sound very similar to DNA polymerase. It's just that this one polymerizes or creates RNAs. So the RNA polymerase reads something called the promoter. The promoter is just a section of DNA that signifies to the RNA polymerase, hey, start here. So once it recognizes that there's a promoter, the RNA polymerase will start to begin its synthesis. Then the next phase is called elongation. And this is when the RNA polymerase just continues to start to add the nucleotides onto the chain. So it continues to add. And as it's adding and growing the chain, the RNA polymerase can naturally separate the RNA, the DNA strands from each other. Oh. Hey, I'm talking to myself, sorry. Okay, and so as it separates, the base pairs are separated from the DNA strands, but once the RNA polymerase passes, the DNA will just snap back together. So when the RNA strands are created, it doesn't destroy the structure of the original DNA strand. And then the last phase is termination. This is when the RNA polymerase reaches the end, and it reads there's a special code at the end that will allow the RNA polymerase to know that it should stop coding. And then it will just detach from the DNA strand and also and releases the mRNA strand as well. Just to clarify, mRNA stands for messenger RNA. That's the kind of RNA that brings the DNA, the genetic message, to create proteins. So there are some modifications that happen to mRNA before it can leave the nucleus. And this is very specific to eukaryotic cells. So for prokaryotic cells, remember, they don't even have a nucleus, so they don't go through any modifications. As soon as the mRNA is created, it gets translated by the ribosomes. But here, there has to be some protection of the mRNA before it can leave the nucleus. So this is a picture of the kind of modifications that happen. The first one is the 5' cap that's kind of stuck at the beginning of the RNA strand. And what it does is it protects the mRNA from getting degraded by exonucleases. They're these proteins that will try to destroy anything that they think is foreign mRNA. And so if it doesn't have the 5' cap, then this newly formed mRNA will get destroyed. Also, this 5' cap is used by the ribosomes to recognize whether or not this is something that it should be translated. And the other thing that is attached is the poly A tail, which is just a series of adenine, which is just the A nitrogenous or nucleotide within that nitrogenous base. And so those are added at the very end. And so this just continues to further protect the mRNA from getting degraded. And it also, when the ribosome is reading this mRNA, when it reaches the poly A tail, it knows to stop coding. The last thing that happens is this mRNA has a mixture of DNA that's useful and mRNA that they don't actually need for the protein. So the exons are the sections that are the genes that will actually be expressed, and introns are just the intervening sequences between exons that don't really do anything. And so before this leaves the nucleus, there will be proteins that come and cut out this part that is not necessary and just pushes together only the important parts, which are the exons. And notice that what's considered an exon and what's considered an intron can be different for each protein. And this is how we can have so many different proteins with minimal amount of DNA. All right, now we're going to be talking about the translation part. There's something called the transfer RNA, which is tRNA, and this reads the codons on the mRNA and carries a very specific corresponding amino acid. So there's a part of the tRNA that has something called the anticodon, and it's just the complementary base pairs for the codon on the mRNA. And so that when they base pair and it kind of clicks, then the amino acid will be added to the growing amino acid chain for the protein. Notice that in terms of the structure of tRNA, it's a single strand of RNA, and so because it's single-stranded, it actually can have a 3D structure where it base pairs with itself, and so it looks kind of like a clover leaf. The bottom part has the anticodon, and the top part carries the very specific amino acid that corresponds to this codon. Ribosomes are also made of RNA, but it's called rRNA, which stands for ribosomal RNA. Getting more in depth with the structure of RNA, of ribosomes, they kind of look like a hamburger. It has like this top part and this bottom part. And so the mRNA kind of gets fed through the ribosome, and then there's 3 slots inside of the ribosome where the tRNAs kind of come and sit and read the mRNA that's passing through. So just to review all the different types of RNA that we mentioned in this lecture, there's mRNA, which is messenger RNA. That's the important one that carries the structure, the information for DNA to become proteins. So that's a messenger RNA. tRNA is the special kind of like cloverleaf structure that comes, and it reads the codons on the mRNA and brings the appropriate amino acid. And then rRNA, ribosomal RNA, is what makes up the material of the ribosome. All right, thank you so much for watching. I'll see you guys later. Bye.